During the last two lectures, we talked about the adaptive mechanisms in response to stress, and we talked about reversible and irreversible injury, causes of cell injury in general. During this lecture, we will talk about the mechanisms of cell injury. There are several general principles regarding the mechanisms of cell injury. We have multiple mechanisms, but those mechanisms may act all together at the same time. So you should not look at them as separate mechanisms because they may all cooperate in the same scenario, or the same cause of cell death can lead to cell death by different mechanisms. Now, there are several principles that the cellular response to injury depends on different things. It depends on the type of cell injury. Is it ischemic injury? Is it trauma-induced injury? Is it a chemical injury, or what? It also depends on the duration and the severity. For example, if we talk about an ischemia, ischemia for a long period of time will cause damage and necrosis. However, ischemia for a short period of time may not lead to necrosis. And it also depends on the cell type. The severity of injury, a low dose of a drug or a toxin may not cause injury as a large dose of this same toxin. And the consequences of injury depends on the type of cell, the status of the cell, and the adaptability of these cells. For example, if we talk about ischemia to a skeletal muscle and ischemia to cardiac muscle, the type of tissue affects the consequences, because an ischemia to the heart of 20 to 30 minutes may lead to myocardial infarction and necrosis. However, we need 2 to 3 hours in the skeletal muscles of the lower limbs, for example, to lead to similar infarction or necrosis. So the type of the cell affected by the injury plays an important role. In the brain, for example, even very few minutes of ischemia can lead to injury. The status of the cells, are these cells diseased from the start, or they were normal looking cells, and the ability to adapt, for example, for periods of hypoxia, some tissues can adapt, but some tissues are highly susceptible to hypoxia. The genetic makeup of the injured cell is very important, because sometimes different cells in different individuals may develop different consequences, for example, to the same toxin. For example, different individuals may have different types of cytochrome P450 enzyme in the liver. So some patients may respond to this toxin in an exaggerated manner, and some patients may respond by minimal effect. This depends on the genetic makeup of the injured cell. This is also important in cases of drugs. Some patients may develop a reaction to a certain dose of a drug that is different from other patients. This is related to the different genetic makeup of the injured cells, and that is what we call it pharmacogenomics. And it's an important field to study nowadays. So the cell injury results from functional abnormalities, because the injured cell will be non-functional at some point, and due to biochemical abnormalities that occurs within the cell itself. And these abnormalities may affect multiple cellular components, not only one cellular component. Now, these biochemical mechanisms of cell injury, they are summarized in the following picture. We have different causes of cell injury, as we studied from the first lecture, different stimuli for cell injury, and at the same time we have different mechanisms, and the stimuli for cell injury with the mechanism of cell injury will determine whether the cell will die by necrosis or will die by apoptosis, these two important patterns of cell death. So we can subdivide the mechanical or the principal biochemical mechanisms. We have many others, but these are the principal biochemical mechanisms of cell injury. First mechanism is by hypoxia and ischemia induced, which will lead to decrease ATP production, because as you know every cell needs oxygen to produce ATP from ADP, and this ATP هو energy source for the cell. So when we have hypoxia and ischemia, the mitochondria will not be able to consume the oxygen to produce ATP, so we have a reduction in ATP. And as you know, every energy dependent function in the cell that uses ATP, starting from the membrane pumps to the ribosomes, protein production, etc. All of these will be decreased, this leading to necrosis at the end. Multiple injurious stimuli also can act by increasing the production of reactive oxygen species اللي بنسميها free radicals اللي هي بتعمل damage للlipids والبروتينز والnucleic acids and can lead to necrosis. Less severe injury like mutations, stress, some infections may lead to accumulation of misfolded proteins in the cell, and this accumulation will trigger the death by apoptosis. This is true with certain viral infections, for example. Radiation, sun-induced damage to the cell, which can lead to DNA damage للمادة الوراثية in the nucleus, also will trigger death by apoptosis. Certain infections, more severe infections, immunologic disorders like allergy, for example, autoimmune diseases will lead to inflammation and generation of toxic molecules, which can lead to mainly necrosis in addition to some apoptosis. So these are the principal biochemical mechanisms, and we will study each one in details. So the first mechanism of cell injury is by hypoxia and ischemia. As you know, oxygen is needed by all cells to produce energy in the form of ATP by oxidative phosphorylation that takes place in the mitochondria. This is the efficient pathway of producing energy in the cell. So a reduction in oxygen supply to the cell, whether due to ischemia, which is the most common cause of hypoxia, or other causes of hypoxia, leads to depletion of cellular ATP. وبالتالي there will be failure of the energy dependent pathways in the cell. واحدة منها هي membrane transport by the sodium potassium bomb, and protein synthesis in the ribosomes, lipogenesis, production of lipids, and phospholipid turnover. So all of these pathways will cease or will decrease in an attempt for by the cell to withstand the period of hypoxia. Some cells which have the ability to withstand more periods of hypoxia, like the liver cells and skeletal muscle cells, because they store glycogen, they can use this stored glycogen in a process called anaerobic glycolysis. That's to generate ATP without the need for oxygen, but it is a less efficient way of energy production, because the amount of ATP produced in anaerobic glycolysis is much less than the amount produced in oxidative phosphorylation. But it is an adaptive mechanism by some cells, like the liver cells and skeletal muscle cells, for the periods of hypoxia. That's why we find that skeletal muscle cells and liver cells need a much longer time of hypoxia حتى نوصل للنكروسز. مثلا skeletal muscle cells ممكن نحتاج ل 2 to 3 hours, for example, of ischemia and hypoxia for these muscle cells to die. So that's because of the anaerobic glycolysis, versus other cells or other tissues that cannot withstand any period of hypoxia, like the brain cells and the heart cells. Their functions start to cease after a few minutes of hypoxia, that the heart will stop beating after 1 to 2 minutes of hypoxia, and we need 20 to 30 minutes of hypoxia for myocardial infarction to take place. So the consequences of hypoxia and ischemia, what I want to say to you, depends on the type of tissue, depends on the duration of the hypoxia and ischemia, and the severity of the event, the severity of hypoxia. The net effects of hypoxia on the cells can explain to us the consequences of cell injury and the changes, ultrastructural changes and microscopic changes that we see in these cells due to the cell injury. The first effect of hypoxia is the reduced activity of the membrane ATP-dependent sodium-potassium bonds. This will lead to sodium and water accumulation inside the cell, and this will lead to the cellular swelling that I talked about in the previous lecture. The second effect is lactic acid accumulation, and this happens in the tissues that undergo anaerobic glycolysis to produce energy. Lactic acid accumulation will lead to the reduction in the pH of the cell, and reduced pH will lead to failure of different enzymes in the cell. Also, reduced oxygen supply will lead to disruption of ribosomes from the rough endoplasmic reticulum, and this will lead to decreased protein synthesis. There is some evidence that hypoxia will lead to accumulation of reactive oxygen species. It is not well understood how this happens, but there is a role for reactive oxygen species effect in the cases of hypoxia. The effect is more if the tissue that is affected by hypoxia يتعرض إلى reperfusion, as we will discuss in the coming slides. So reactive oxygen species are produced in a much larger amounts in the ischemia reperfusion injury, but it is produced in hypoxic injury. Also, hypoxia will lead to damage to mitochondrial and lysosomal membranes, and this damage to the whole membranes of the cell will lead to, at the end, to necrotic cell death. Another mechanism of cell injury is what we call the ischemia reperfusion injury. كلنا نتوقع إنه أي ischemic tissue لما يصير له reperfusion, for example كان في blockage في البلد vessel وبعدين صار relief of the obstruction in the artery وصار في reperfusion to the tissue، يعني رجع تدفق الدم. احنا بنتوقع إنه التشو ينتعش ويرجع back to normal، لكن what happens in certain conditions that the reperfusion to an already ischemic tissue but viable tissue will lead to a paradoxical effect of cell injury due to several mechanisms. This is commonly occurs sometimes after myocardial infarction and cerebral infarctions, and will lead to substantial tissue injury after the reflow of blood. The mechanism of ischemia perfusion injury is first by increased production of reactive oxygen species, and this occurs due to the inefficient use of the oxygen by the damaged mitochondria of the injured cells. This damaged mitochondria will lead to incomplete reduction of oxygen in the oxidative phosphorylation steps in the mitochondria, and this will lead to production of reactive oxygen species, and the infiltrating new leukocytes will add to these reactive oxygen species by liberating more of these free radicals. The other mechanism is by increased inflammation. The inflammation is already there by the necrosis of the ischemic cells, but we have an influx of leukocytes and plasma proteins and complement components from the blood that comes to the site of injury. This will lead to increased inflammation and increased damage, complement activation, and the net result is an injury, exacerbation of the injury to these cells. Another mechanism of cell injury is the oxidative stress. Oxidative stress is the cellular damage and cellular abnormalities induced by the reactive oxygen species, which belong to a group of molecules called free radicals. What are free radicals? Free radicals are species, chemical species, that have a single unpaired electron in their outer shell. This unpaired electron makes them extremely unstable with high energy, that if they react with any other organic or inorganic molecules, they will convert them to free radicals too, and they will induce damage to the cellular proteins, cellular lipids, and cellular nucleic acids. When these reactive oxygen species or free radicals produced in chemical injuries, like in carbon tetrachloride injury, in radiation injury induced by X-rays and the UV light from the sun exposure, hypoxia, as I said in the previous slide, cellular aging, inflammation, and ischemia, reperfusion injury, which we already discussed. So in all of these scenarios, reactive oxygen species are generated, and the generation of reactive oxygen species will lead to a production of more free radicals and the propagation of the reactions. Now, the net effect of reactive oxygen species produced in redox reactions depends on the rate of production and the rate of removal, because normally small amounts of these reactive oxygen species are produced in all cells during the redox reactions in the mitochondria, during the reduction of oxygen to produce water. But because these are very small, these are very small amounts, and they are short-lived, so they undergo spontaneous decay, there will be no net result of damage to the normal cells. But if their amount is increased, there will be damage. Normally, the oxygen is reduced in the mitochondria in the oxygen reduction reactions into the superoxide ion. And this superoxide is a free radical. And then it is converted into hydrogen peroxide, H2O2, which is also a reactive oxygen species that can cross cell membranes. This conversion from superoxide to hydrogen peroxide is performed by the action of the superoxide dismutase enzyme. Then the hydrogen peroxide H2O2 is also reduced into the hydroxyl radical OH. The second pathway of reactive oxygen species production, in addition to the redox reactions in the mitochondria of all cells, is the production of reactive oxygen species in phagocytic leukocytes, that are the neutrophils and the macrophages during inflammation, in an attempt to kill the microbe or the phagocytosed bacteria, for example. In these situations, oxygen is also reduced into the superoxide, and the superoxide is converted into H2O2 by the action of the superoxide dismutase enzyme. Then the H2O2 is converted in these macrophages and in neutrophils into the hypochlorite, which is the bleaching agent اللي موجود نستخدمه كلياتنا في بيوتنا، وهو يعني يعتبر microbicidal agent. It can kill bacteria and other microbes. This action of conversion of H2O2 into the hypochlorite is performed by the enzyme which is present in leukocytes called myeloperoxidase, or MPO. Now we come to the removal of these free radicals. The mechanisms that remove free radicals will limit the damaging effect of free radicals on the cells. First one is spontaneous decay. For example, the superoxide can spontaneously be converted to the hydrogen peroxide, but the main enzyme that performs this function is the superoxide dismutase, which converts the superoxide into the H2O2. Now the H2O2 is converted into water by the action of glutathione peroxidases. The most common of them is glutathione peroxidase 1, and also by the action of catalase, which is one of the most active enzymes known. It will transform millions of the particles of superoxide into of H2O2 into water. So glutathione peroxidase and catalase, both of them will perform the same action, which is conversion of H2O2 into water. Other scavengers for the free radicals, the endogenous and exogenous antioxidants like vitamin E, vitamin A, vitamin C, and the beta carotene. These are antioxidants, so either they block the production of free radicals or scavenge them as soon as they are produced. The net result of production and removal of these free radicals will decide the extent of the damage they perform in the cell. Now, this picture summarizes what I have said about the production and removal of free radicals within the cell. As I said, that if the cell is exposed to injury, like radiation, toxins, reperfusion injury, for example, inflammation, whatever type of injury that we talked about, hypoxia, this will lead to production of more amounts of reactive oxygen species in the mitochondria, and the other pathway is in the leukocytes. Now, in the mitochondria, the oxygen reduction reactions that occurs normally produce minimal amounts of these free radicals that are removed immediately, and they will not lead to the net damage and the pathologic effects of reactive oxygen species. But in disease conditions or in injury circumstances, the amount of free radicals will be more. So first, oxygen is reduced into into a superoxide, and this superoxide is converted into H2O2 by the action of superoxide dismutase, the first pathway in removal of these free radicals. Then the H2O2, the hydrogen peroxide, is also converted to either converted to another free radical, which is hydroxyl radical, by the action by the aid of iron in a Fenton reaction, or if the cell wants to limit the damaging effect of H2O2, it will be converted into water by glutathione peroxidase and the catalase enzyme. So these actions here leads to removal of free radicals, but if the damage is severe, the تفاعلات تكوين الفري radicals will increase باتجاه تكوين more free radicals, which will lead to different pathologic effects like lipid peroxidation of the lipid اللي موجود في البلازما membranes leading to membrane damage, protein modifications leading to breakdown and misfolding of proteins and accumulation of misfolded proteins in the cell, also will lead to DNA damage leading to mutations in the DNA. So these are the net pathologic effects of reactive oxygen species formation. So what are the effects of these reactive oxygen species, and how are they involved in the mechanism of cell injury? These reactive oxygen species first will do lipid peroxidation for the lipid residues in the membranes. This peroxidation happens when the reactive oxygen species react with the lipids present in the plasma membranes, lysosomal membranes, and mitochondrial membranes, leading to the production of peroxides. These peroxides themselves are unstable and will mediate a destructive process to these membranes. Second, the reactive oxygen species will cause cross-linking في proteins. And this cross-linking of proteins will lead to degradation of these proteins and fragmentation with loss of enzymatic activities, in addition to improper folding of these proteins, leading to accumulation of misfolded proteins in the cell, which may also predispose to or activate the process of apoptosis. Third, these free radicals may cause DNA damage at the level of the mitochondrial DNA and the nuclear DNA, leading to single-strand breaks in the DNA. And these breaks or this damage will mediate apoptosis, will mediate aging, and malignant transformation in certain instances. The last effect of these free radicals is the killing of the microbes, and this is the beneficial role of these free radicals during inflammatory reactions. We need this effect. So at the end, reactive oxygen species are produced normally and in small amounts in normal cells in physiologic conditions. However, in pathologic conditions, they are produced at larger amounts and leading to the catastrophic effect to the cell. Another mechanism of cell injury is the cell injury caused by toxins. Toxins are environmental chemicals and substances produced by infectious pathogens. These toxins may act directly, so we call them direct acting toxins, and may act by their metabolites after they are converted to metabolites in the body, not by themselves, and we call them latent toxins or indirect acting toxins. So let's start with the direct acting toxins. They act directly by combining with a cellular organelle or a critical molecular component in the cell, like a membrane, a phospholipid, for example. And the best examples on direct acting toxins are the mercuric chloride poisoning اللي هو بيجي نتيجة ingestion of contaminated seafood and the chemotherapeutic agents which are used to treat cancer, antineoplastic drugs. These both of them will act directly by reacting mainly with the cellular membranes, leading to damage of these membranes. They react, for example, with the sulfhydryl groups of membrane proteins, so they will inhibit the ATP-dependent transport, leading to cellular swelling and so on. So the effect is direct on these cells. Now, the latent toxins or the indirect acting toxins are not intrinsically active by themselves. They should be converted to reactive metabolites that by themselves cause the intended damage and act on the target cell. And this transformation usually takes place in the cells that contain the cytochrome P450 enzyme in their smooth endoplasmic reticulum, that like the liver cells. So these latent toxins, they act on target cells, not all the cells are affected. Only those that use the cytochrome P450 enzyme. The damage they exert is mainly by the formation of free radicals, which lead to, as we said before, to membrane phospholipid peroxidation. The best examples on these latent toxins are carbon tetrachloride, CCl4, which is converted into a free radical CCl3, and acetaminophen, اللي هو it's an analgesic or painkiller that you all know about it. So the effect of these chemicals is by first membrane lipid peroxidation, leading to damage of these cellular membranes and organelle membranes. It will damage the endoplasmic reticulum membranes, leading to swelling of the endoplasmic reticulum, decline and detachment of ribosomes from this endoplasmic reticulum, with a consequent decline in the synthesis of proteins and enzymes, in addition to decreased synthesis of apoproteins. The apoproteins especially they are produced in the liver, وهم المسؤولين عن the binding to the lipids to carry them out of the hepatocytes for their secretion. So when we have a decrease in apoprotein synthesis, lipids and triglycerides will accumulate in the liver, causing a fatty liver. And this is one of the signs of toxicity of CCl4. Third, we have also mitochondrial injury due to mitochondrial membrane injury. And this will lead to reduction in ATP production, reduction in ATP-dependent membrane transport, cellular swelling, and cellular death. Again, this picture here shows you the mechanism of CCl4 toxicity, and the same applies to acetaminophen. The CCl4 is converted in the liver in the smooth endoplasmic reticulum by the cytochrome P450 enzyme into a free radical called the CCl3. And this CCl3 will lead to lipid peroxidation of the membranes, including the plasma membranes and organelle membranes, including the endoplasmic reticulum membranes, mitochondrial membranes. Plasma membrane damage will lead to membrane permeability, damage to the ion transporters. Membrane permeability بتزيد, sodium and water enters to the cell, leading to cellular swelling and mitochondrial swelling. The mitochondrial swelling and the mitochondrial damage leads to decreased ATP synthesis. In addition, the damage to the endoplasmic reticulum membranes leading to detachment of ribosomes and decreased protein synthesis and apoprotein synthesis, and this will lead to the fat accumulation inside hepatocytes, leading to fatty liver, which is the sign of toxicity for CCl4 and for acetaminophen. Let's move to another mechanism of cell injury, which is the endoplasmic reticulum stress. Now, the proteins are usually synthesized in the endoplasmic reticulum. These proteins, after production or after synthesis, they are folded in a proper way in order for them to function properly. And this folding process needs compounds or molecules called chaperones. Now, when these proteins are folded in an improper way, we call them misfolded proteins. They are targeted for ubiquination, and this will lead to their damage, proteolysis. When a cell produces unfolded proteins in a larger amount, the cell first tries to adapt by activation of the unfolded protein response, or what we call it, the adaptive response. This adaptive response includes the increase in chaperone production in order to fold these unfolded proteins, and it induces the cell to decrease protein translation and increase protein destruction, so that the unfolded proteins will decrease in the cell. Now, if this is failed, there will be activation of the apoptosis by first activation of pro-apoptotic sensors موجودين في السيتوسول من ال PH3 family. We will talk about them in apoptosis, and this will lead to activation of caspases, which are the apoptotic enzymes, and the start of apoptosis by the mitochondrial pathway. So again, as what I have said, this is the endoplasmic reticulum lumen, and this is the endoplasmic reticulum membrane, and this is the cytosol. When we have increased amounts of misfolded or unfolded proteins, this is called a mild, in small amounts, this is called a mild distress. In this situation, the cell will try to adapt. We call this an adaptive unfolded protein response. How this adaptation will take place is by increasing synthesis of chaperones in order to fold these proteins properly, and reduce synthesis of proteins and increase degradation of proteins. So the net result is the reduced load of these misfolded or unfolded proteins. Now, if the amount of these unfolded proteins is too much, we call this a severe stress. يعني بتعمل ضغط عالي على endoplasmic reticulum, severe endoplasmic reticulum stress, and this will not activate an adaptive response. لا، في هاي الحالة it will activate an apoptotic pathway بنسميه mitochondrial pathway of apoptosis, activation of PH3 sensors in the cytoplasm, activation of caspases, and the end result is apoptosis of the cells containing these amounts of unfolded or misfolded proteins. What are the causes of protein misfolding that will lead to this type of cell injury and so activation of apoptosis, as I said in the previous slide? First, gene mutations leading to production of proteins that cannot be folded properly. Second, aging. With aging, the capacity of the cells to fold the proteins will decrease. Ability to produce chaperones will decrease. In certain infections, especially viral infections, will lead to the accumulation of viral proteins within the cell, وهي بتعمل stress على الخلية. Increased demand for secretory proteins like the insulin. في الناس اللي عندهم insulin resistant states، الجسم بيتعامل مع الانسولين الموجود وكأنه مش موجود لأنه في resistance to this insulin، فالبنكرياس بيستجيب by producing more insulin, but this insulin accumulates in the cells لأنه بصيرش في capacity عالية to fold this insulin properly, so it will accumulate in a misfolded form in the pancreatic cells, leading at the end to their damage and death by apoptosis. Sometimes ischemia and hypoxia may lead to unfolding of or accumulation of unfolded proteins, and some neurodegenerative diseases, especially Parkinson's disease and Huntington disease and Alzheimer disease. طيب how does this protein misfolding causes the disease؟ احنا حكينا ال accumulation of misfolded proteins may turn on the apoptosis pathway and leading to cell death by apoptosis. بس مش دائما ال misfolded proteins بتعمل ال disease by apoptosis لحاله. ممكن برضه by other pathways. إما by deficiency of an essential protein due to a degradation of this protein, like in a disease called cystic fibrosis. There is a genetic mutation in a protein that is needed as a membrane transporter, so there will be production of an unfolded protein leading to its degradation. So we will end with a state of deficiency of this transporter in the cell membrane, and كل الأعراض الناتجة من cystic fibrosis is due to the deficiency of this protein. Sometimes it will cause the disease by inducing apoptosis of the affected cells. السيلز اللي بتكون بيتراكم فيها misfolded proteins، وهذا mostly بنشوفه في neurodegenerative disorders like Alzheimer's disease, Huntington disease, and Parkinson's disease, and also in type 2 diabetes. As I said in the previous slide, insulin is produced in a larger amounts في حالات insulin resistance in type 2 diabetes, and this insulin will accumulate in the cells بشكل يفوق the capacity of the pancreatic cells to fold this protein properly, so it will turn on the damage to these cells by apoptosis due to the accumulation of misfolded proteins. Also, protein misfolding lead to accumulation of improperly folded proteins in the extracellular matrix, as in cases of amyloidosis. So these are the three mechanisms by which protein misfolding leads to the disease process. Another mechanism of cell death and of cell injury is the DNA damage. DNA damage can be implicated on cells due to radiation, to chemotherapeutic agents, to generation of reactive oxygen species in the cells, as we stated before, and mutations. Now, any damage to the DNA that occurs is sensed by the p53 gene, leading to p53 activation. What is p53? It is بيسموه guardian of the genome، وهو عبارة عن tumor suppressor gene. أي damage في الدي ان اي بيخلي هذا ال p53 ينتبه له، بيصير له activation، فبيعمل arrest للcell cycle at the G1 phase، يعني بيحكي لها استني stop in order for the DNA damage to be repaired. If the repair occurs بترد تمشي الخلية في بقية السيل سايكل، لكن if the repair of this damage is impossible ال p53 بيخلي الخلية تروح للapoptosis، بصير لها activation of apoptosis عشان تختفي. Now إذا المريض عنده أو البيشنت عنده p53 mutation، إذا أي خلية تعرضت ل DNA damage، these DNA damaged cells will replicate, they will continue to replicate، ما في إشي يمنعهم، وهذا اللي بيؤدي ل neoplastic transformation. فهذا اللي بيصير لما مثلا إحنا بنتعرض ل DNA damage اللي بالsun exposure. الsun exposure بيعمل DNA damage في الخلايا الجلد for example، فهذا بيؤدي ل activation of the p53 gene، فبيعمل arrest للcell cycle في خلايا الجلد عشان ما تتابع ال proliferation of a DNA damaged cell. If this p53 protein is mutated or abnormal، راح نصير نتعرض لسرطانات الجلد زي البيزل سيل كارسينوما و squamous cell carcinoma for example. And another mechanism for the cell injury is the inflammation. Inflammation is the normal reaction of the body elicited by pathogens like microbes, for example, by necrotic cells, by dysregulated immune responses like in autoimmune diseases وتفاعلات الحساسية allergies. لما يصير في عنا inflammation بيصير في accumulation of inflammatory cells, mainly neutrophils, macrophages, and lymphocytes, and these cells produce many products that destroy the microbe, but at the same time they cause damage to host tissues and cell injury. So after studying the many mechanisms of cell injury, we end up with events that are common in cell injury from different causes. If you noticed that most of the causes that we talked about lead at the end سواء من hypoxia لا ischemia لا toxins لا free radicals, they will end at by mitochondrial dysfunction and defects in membrane permeability. So mitochondrial dysfunction, for example, can result from hypoxia and ischemia and toxins and radiation injury. And as we know that mitochondria is the energy factory of the cell, so it is usually involved in both death by necrosis and apoptosis. The consequences of mitochondrial dysfunction is first failure of the oxidative phosphorylation, leading to ATP depletion, and also the abnormal or inefficient oxidative phosphorylation leading to formation of reactive oxygen species. Also the formation of permeability transition pores in the wall of the mitochondria leading to loss of membrane potential and inefficient oxidative phosphorylation. Also, in certain cases, there will be release of cytochrome C, which is usually stored in the mitochondria, into the cytoplasm, triggering the process of apoptosis. So mitochondrial dysfunction is both involved in necrosis and apoptosis. So whatever was the cause of the mitochondrial damage, whether it is decreased oxygen supply, toxins, radiation, this will lead to membrane damage, decrease of the mitochondria with the reduction in ATP generation due to inefficient oxidative phosphorylation and production of reactive oxygen species leading at the end to necrosis. In addition, also mitochondrial dysfunction is involved in the apoptotic pathway of cell death by decreasing survival signals, DNA damage, protein damage to the cell will activate pro-apoptotic proteins and will lead to formation of pores or channels in the wall of the mitochondria, leading to the escape of cytochrome C to the cytoplasm, promoting apoptosis. And regarding the depletion of ATP, one of the most important consequences of mitochondrial damage due to oxidative phosphorylation shut off is the inability of the sodium-potassium pump to function, leading at the end to cellular swelling and endoplasmic reticulum swelling. Also, depletion of ATP will activate anaerobic glycolysis, leading to a lactic acid accumulation, decreased pH of the cell, and clumping of a nuclear chromatin and damage to enzymes and proteins. Also, decreased ATP will lead to detachment of ribosomes and decreased protein synthesis. So this is just a revision to what we already said. The second commonly shared consequence of all the mechanisms, of most of the mechanisms or causes of cell injury is defects in membrane permeability. سواء الميتوchondrial membranes, their damage leads to decrease in ATP production and loss of the membrane potential. Plasma membrane damage leading to loss of osmotic balance, influx of fluids and sodium inside the cell, and leak out of cellular contents. Lysosomal membrane damage leading to release of enzymes into the cytosol, leading to cellular digestion. So this is what you have to know in general regarding the mechanisms of cell injury. Thank you.